I do remember when researchers were first working on liquid biopsies, and everyone said it was not going to work. But they continued their research. And I remember initially, I read things where you'd have to take so much blood you couldn't possibly weed out all of the different things that were floating around in blood and be able to make definitive um, diagnosis and definitive prognosis decisions. And those researchers proved those naysayers wrong. I was first diagnosed 37 years ago. I was a very young person back then, with what was then referred to as a double negative breast cancer because they had not yet invented HER2 or discovered it. I had positive nodes and frankly, not the best prognosis. They sort of threw the kitchen sink at me, which is what they did back in those days, and it apparently worked. However, 15 years later, I got a second breast cancer in the other breast, totally different. This one was estrogen receptor positive, but was node negative. Unfortunately, I was diagnosed just before the development of those assays that determine whether or not you needed chemotherapy or not, like an Oncotype DX. So I got more chemotherapy. And then 9 years after that, I got a third breast cancer, which was a DCIS in the other breast. So I kind of alternate, and have had, they were all early stage, but I had to go through this 3 different times. But the experience actually was interesting because it gave me a perspective how things have actually changed a great deal over the last 37 years in how we screen for this disease. And certainly treatment has changed very significantly. And it has given me an appreciation of how clinical trials and research has made things a whole lot better in many respects. I think one of the things I'm most excited about this new technology is really learning a lot about the tumor that the clinician is dealing with in a relatively, well, minimally invasive way. One of the problems I've always felt about traditional biopsies, where you're taking a piece of tissue, is that you're only looking at one small bit of that tumor under a microscope. And you can miss things because tumors, by their very nature, have a great deal of heterogeneity. And this way, you're getting maybe a more complete picture of what you're dealing with genomically from their DNA. And as a result, I think you can get much more personalized treatment and much more targeted treatment if you really understand what you're dealing with. So, from an early stage standpoint, you know, better determination whether or not neoadjuvant therapy has worked. It might be able to detect minimally residual disease that might be helpful. Um, and clearly there's a lot of work in using it for screening in terms of understanding whether or not you might have a very early something that might replace ultimately mammography or other forms of screening. Of course, the biggest issue there is with any new technology is making certain that what it's measuring is true. There's an issue both with false positives and also with false negatives. And this whole issue of specificity and effectiveness is still early, particularly in the screening area. But I do think it could ultimately be very exciting. Clearly, there has been strong data that shows that using liquid biopsy, particularly in the treatment of metastatic breast cancer, does have utility. And I do think that that is something that we're going to see more and more in practice and that patients are going to be asking for. Um, what I would urge all patients is to consider going into clinical trial. I mean, clinical trials are the way that we make advance. And the only way that we're going to make liquid biopsy better and and better for those that come after us is to get the data and to learn and figure out how to best use it. and to improve the technology. And the best way to do that is to have um, a diverse group of people be willing to participate in a clinical trial so that we really know how it works, what doesn't work, what will work better, and to get smarter about this.